Okay, so, how y'all doing? You ready to do some physiology? Excellent. It is very cold in here today. I don't know what's going on. But we press onward. So, let's warm ourselves with the beautiful fire of neurotransmission. Let's get excited. So, let's talk about how signals get sent in your nervous system, right? So, nerve impulses, tiny, brief electrical spikes that occur within neurons, and this is how you send information. That was it. Cool. But let's do some detail, right? It works by moving chemical signals, the chemical ions, across the cell membrane in a wave called an action potential. So each neuron only sends one kind of impulse, and only go one direction. It sort of used to be thought, or at least theorized, in early physiology of the nervous system, that it's sort of like a tube where stuff can go both directions, and you send information, and then you send like motor signals back the other way. But actually, it's set up so that it only goes one direction, only sends one kind of signal, and it's an all or nothing signal too, right? It's on or off. You can only be firing or not firing, right? So the way you get different intensities of sensation and movement and whatever it is you're trying to communicate is just through frequency of firing, right? But if you need to communicate with somebody by turning a light bulb on and off, you can't, like, you don't have a dimmer switch where you can like turn it up bright or only up a little bit. So you can still communicate different intensities with frequency, right? If you just need to like say hi, you just like flick the lights off on and off a couple times. And if there's a serial killer in your room and there's an emergency, you like flick the lights a whole bunch of times really fast, right? Your nerves do that same thing. Even though you don't have a dimmer switch on your nerves, you use frequency as a code for intensity. So, you communicate between cells, between neurons, with neurotransmitters that move across the gaps between the cells, called the synapse. And the synapse is the gap between the presynaptic neuron, the terminal buttons there, and the dendrites of the next postsynaptic neuron, right? The synapse is the gap, right? You know, gets on my nerves? Myelin. So, how an action potential works. A resting neuron that's not doing anything is polarized, right? There's more positive ions outside and more negative ions inside. So there's this difference between the outside of the cell and the inside, a sort of resting electrical charge. And when you need to send a signal that starts up in the cell body and then travels down the axon, you get this rapid depolarization as the impulse passes, right? Where the ions move into the cell and out of the cell to create a gradient that goes the other direction, and then like moves down to the next one and keeps going, right? So we're going to watch a little video talking about exactly how this works.
Electricity as you may know it. I've said before that a nerve impulse is an electrical signal that travels along the neuron, but it's not quite electricity as you may know it. Electricity is the flow of electrons, but in a nerve impulse we use the word potential to describe the movement of charged particles across the membrane of a cell. The charged particles flow like a wave down the axon, moving in and out of the cell. The movement is mostly of positively charged ions, not negatively charged electrons like you might imagine. Let's look even closer at the cell membrane. At rest, when a neuron isn't firing, there's a high concentration of sodium ions outside of the cell membrane, and also a high concentration of potassium ions in the cytosol of the cell. The sodium and potassium channels are gated and closed. Because of the difference in number of sodium and potassium ions, as well as the presence of negative ions like chlorine, there's an overall negative internal charge of negative 70 millivolts. The beginning of an impulse is marked by depolarization. This means that the voltage-gated sodium channel is opened and the sodium rushes into the cell. The influx of positive charges quickly shifts the electric potential of the membrane, so that now the outside is more negative and the inside of the cell is more positive. This change in polarity will close the sodium gate. Nearby is the voltage-gated potassium channel, which will now open and allow potassium ions to flow out of the cell. The movement of these positive ions will repolarize the cell, allowing the cell to have a negative internal charge relative to the positive extracellular charge. The change in polarity will also close the potassium gate. At this time, you might think that the cell is ready to receive another signal, but it can't. If the sodium gates opened, the sodium ions would not move. In order for ions to move through their channels, they have to be able to move down the electrochemical gradient. The positive sodium ions aren't attracted to the positive environment outside of the cell, so they won't move. Another signal can't be sent until the sodium and potassium ions have moved back to their resting potential positions. But this means moving against concentration gradients, which means energy must be used. During the refractory period, the sodium-potassium pump will move 3 sodium ions out of the cell and 2 potassium ions into the cell with 1 ATP molecule. This must happen constantly and rapidly, so the brain uses a significant amount of your body's energy. In a matter of milliseconds, the sodium-potassium pumps will restore resting potential and another action potential can move through. Nerve impulses are actually waves of action potentials, which means ions are flowing in and out of the cell in a wave-like pattern. Because of the refractory period, where sodium ions will not flow out of the cell by diffusion, action potentials can only move in one direction. They begin at the dendrites, and they end at the axon terminals. This wave-like region where the action potentials are flowing are referred to as the local current.
Okay, there's a bit more she talks about, but that's enough for us for right now. So that's how it goes, right? You got sodium and potassium channels, right? You think of it like there's a gradient, like you're sitting on top of a hill ready to roll down, and once the gate opens, all the stuff just like rolls down the way it needs to go, and then you need to like have a pump, a sodium potassium pump that pumps it back out where it's supposed to go, like you get on the ski lift that takes you back up to the top of the hill, right? Which takes energy. And this is what a lot of your brain's energy use is doing, right? That we talked about how much energy your brain uses, how much glucose and stuff it takes. Make it into ATP, benzene triphosphate, which is sort of the raw material your brain breaks stuff down into for energy. And you spend it like pumping these ions back out of the cell or back into the cell where you can set up for another action potential, right? So channels of the membrane allow positively charged sodium ions in and out positively charged potassium ions to restore the polarization. So at the synapses, we get down to the end, and like we talked about with the shape of the neurons themselves, a synapse can be at lots of different places, right? So you can have some like terminal buttons that come not necessarily to these long dendrites out here, but down closer to the cell body, or you can have a shaft synapse, what it's called, or like a little spine synapse, or axonic synapse, right? That like comes in and talks to it right before the terminal buttons, which is kind of cool. So synapse gets very, very close, but doesn't touch. And neurotransmitters cross the gap in 2 milliseconds, which is a 500th of a second, which is very, very fast for like chemicals pushing across. But because it's a very, very small gap, it happens very quickly, and the stuff diffuses very quickly, which is what allows us to think at the speed that we do and feel at the speed that we do, right? So one neuron can be in contact with lots of other neurons. It's not necessarily one-to-one. We talked about some last time. And the impulses that it sends can either excite or inhibit the next neuron from firing, right? So you got one neuron that's going to fire, and it's got other neurons coming into it, and depending on which ones are talking to it right now and how much, it's either going to be telling it to fire or not fire, right? Or to stop firing, depending on what the neurotransmitter is and the sort of information that it holds. Does that make sense? Cool. So this is sort of a nice close look down here at the terminal buttons by the synapses. There's a lot of information in here, but we're gonna sort of hit the high points, right? In this like big crazy graphs. You can sort of see like terminal button down here, and then the synapse, that little tiny gap, and then the dendrite of the next neuron. So several steps here, and you've got the presynaptic axon. The action potential gets here, and so suddenly we have like a positive charge in here where there was a negative charge before. And waiting around in here, there are these little pods that have neurotransmitter in them. And positive charge causes these little pods to like flow away and fuse with the end of the cell wall here. And when they do that, they dump their neurotransmitter out into the synapse, right? Or rather, the action potential causes these voltage-gated channels to open, and Ca2 plus comes in. That's where you get the positive charge, and it makes this stuff dump out. That creates a lot of intercellular calcium 2 plus in here, which makes these vesicles dump out their neurotransmitters. It's called exocytosis, right? This process by which these like bump into the end, fuse with it, and spit out their neurotransmitters. And then those neurotransmitters diffuse across the synapse and fit into little binding spots on these receptors, these gates in the next neuron, which causes them to open and let our old friend the sodium ions in, which starts a new action potential, right? In the postsynaptic cell. Right? And so that's it can also, depending on the neurotransmitter and what it is, if it's like an inhibitory neurotransmitter, it might cause these to close up and stop letting sodium in, and cause the neuron firing to stop, right? This is sort of how it goes, right? The calcium flows in, the action potential, and these open, and causes these to dump out. So let's talk about direct and indirect gating for a minute. So as I just said, the neurotransmitter floats across the gap, and we start the process over because it sticks in this gate and causes it to open, or tells it to close, right? Depending on whether it's excitatory or inhibitory. But it's not always that way. There's something called direct gating, which is that, right? The neurotransmitter fits right into this channel molecule, it's the key, that's the lock, it opens it up. Simple. But there's also this other thing that happens called indirect gating, where the receptor isn't on the gate in the next neuron, it's a totally separate molecule sitting over here, right? There's this, so the lock isn't on the door, the lock is somewhere over here in the wall, and you stick the key in the lock, and that causes some other stuff to happen, and then a guy goes over there and opens the door. That's basically indirect gating, right? This receptor stimulates a GTP binding protein, and that activates what's called a second messenger cascade that causes the channel to open, right? That like this, if it's in the neurotransmitter fits in this receptor, and that talks to this G protein, releases GTP, adenyl cyclase, and CAMP, and that talks to PKA here, and this floats over and gives an effector function, and that's what opens the gate. Okay, great. Basically, instead of unlocking the door, you stick your key in the wall, and the wall talks to this other thing, and the light goes on, and that guy goes over and opens the gate. So you might be thinking, that sounds really complicated, right? That like, if you're God and you're in charge, and you're like, okay, this whole process is already pretty complicated. Why don't we just do everything like this? Why isn't it all just direct gating? It seems a lot simpler, a lot easier. Why bother with this whole thing? Why go to this trouble? Which is an excellent question that has a really interesting answer. So we're going to talk about it. So this is sort of what lets individual neurons change over time, which is how you learn things, how you get more sensitive to things, how you grow, neurologically speaking, right? So if you just have the neurotransmitter float over and unlock the door, then you either have the neurotransmitter working or not. You're either firing or not. It's very binary, 1 or 0. That's thinking like a computer thinks. But this whole like weird second messenger system, where by sticking your key in this lock that's over here and turning on the light, and that makes those guys in the little guardhouse start wandering around and unlocking the door for you and stuff. You have this whole second messenger system now floating around in the cell, and you started this whole process within the cell that's not just opening the door, it's doing other stuff, right? That produces a synaptic potential that doesn't just work right then, but keeps working for a little while, right? And this causes causes kinase to translocate to the nucleus, and that can affect the DNA transcription process. You're writing DNA in the nucleus of your cell, in the nucleus of all your cells, and this sort of second messenger cascade can affect sort of the structure of the neuron itself and the way that it perpetuates itself, the way that it grows, right? This is how the whole long chemical process that leads to how a cell stops just being one thing and starts actually anticipating responses in the future, anticipating the sort of the firing that's come across in the past, right? When something fires in your brain, when neurons fire, they're more sensitive to firing that way again, right? When you have thoughts, those thoughts are more likely to come back again. When you have experiences, you get used to those experiences, right? And this is what it is. This whole big thing that I saw some eyebrows go up over, right? Because there's a lot going on, is what makes you not just a computer, right? You're not just ones and zeros. You don't just respond in the exact same way to everything that happens every time. But you grow, you change, and you can thank this sort of indirect gating system for this, right? I like this a lot. I like how when like the really crunchy chemical physiology stuff ties back into something more psychological in this sort of very elegant way. So I really like this. I think this is cool. So this sort of like kinase that's floating around in there, ending up in the DNA transcription process, can alter the excitability of the cell for days or even for longer, right? So, long-term effects from this indirect gating process. Cool stuff. So let's talk about the neurotransmitters themselves. There's 3 major categories here, rather. We got monoamines, amino acids, and acetylcholine, right? Monoamines are things like norepinephrine, dopamine, serotonin, histamine. All these are like monoamine, one amine group. And then you can also have like ones that are full amino acids, like GABA, glutamic acid, aspartic acid, glycine, all work as neurotransmitters, and they're sort of bigger or more complicated, have full amine groups. And then acetylcholine is its own group because it's weird. It's just acetate and choline stuck together. It's not an amino group at all, but it still works as a neurotransmitter. So that's cool. So 3 major categories, and there's a bunch of other subcategories in here that we won't get into today, but this is the big categories. So when you spit out those neurotransmitters into the synapse, how do you get them back? Where do you get this stuff? There's this whole reuptake process where there's this brief recovery delay after a neuron is fired and dumped out its neurotransmitters. There's no more neurotransmitters to get dumped out to talk to the next neuron. So there's this refractory period where like we gotta like wait for a second before this neuron can fire again. It's not a literal second, it's much faster than a second, but at the speeds we're capable of understanding it, we gotta go a little slower, right? This process differs depending on what the neurotransmitter is, but essentially you have enzymes that go out and collect all of the old neurotransmitters, break them down, ship them back into the cell, and then we'll put them back together to get ready for the next time we need to fire, right? So that's called reuptake, and the process is a little different for these different types of neurotransmitter. So let's talk about cholinergic reuptake, which is for acetylcholine. So there's this enzyme called acetylcholinesterase, ACHE, in the synaptic cleft, just sort of like hanging out here. And when acetylcholine comes out here and it's bouncing around, running into the receptors on this cell, your acetylcholinesterase is hanging out there, and it gathers up, catches the acetylcholine, and breaks it in half. And so you have choline and acetate, which are both inert, right? So that you don't just have a lot of like extra acetylcholine floating out there and causing random activations, which you may not want. You only want it activated when it's supposed to be activated. So you got ACHE breaking it apart into its component pieces, and then it breaks it down and like lets them go, the choline and the acetate. And then you have choline transporter picks up those molecules and sticks to them and diffuses back through the neuron terminal back into the cell. And then it's reused in catalyzing more acetylcholine by choline acetyltransferase, CHAT. And so you can sort of see very specific molecules that very specifically have a relationship to acetylcholine, several of them, that are specifically for just this neurotransmitter in this spot, which is pretty cool. The complexity of this gets insane, right? That there's so many different molecules and so many different things that only work with very specific molecules. And so this is the reuptake process, basically for acetylcholine, right? You got this sort of cycle where you're letting it out, and then breaking it, shipping it back in, and then turning it back into acetylcholine. For reuptake for monoamine neurotransmitters, there's basically separate transporters for every specific neurotransmitter type, right? Just like acetylcholine has its own things. There's something called DAT for dopamine, NET for norepinephrine, SERT for serotonin, which just for reference is the thing that SSRIs don't like, right? If you take a selective serotonin reuptake inhibitor, what they do is they basically interfere with the SERT being able to do its job. So when you dump serotonin out here into your into your synapses. The SERT is not there to break it down and send it back in, and so you have more of your own serotonin that you made floating around in there and bumping into the next neuron and causing more activations. Sounds great, right? But a very specific chemical that's a very specific enzyme that's messed with by this drug. VMAT2 repackages the monoamine into neuron vesicles for reuse, right? You have the very specific things hanging out there looking for their specific neurotransmitter and breaking it down and then shipping it back into the cell where it gets repackaged into those vesicles, ready to go. Right? It's vesicular monoamine transporter. And then there's like other chemicals that work for amino acids, right? Neurotransmitters. So GABA and glycine have separate transporters, but amino acids are interesting because we talked about with glial cells of like the way they help with the reuptake process. It's with amino acids that this works, right? So there's channels in these surrounding glial cells that help to like swoop it back up and get it back into the cell, and converts it to glutamine for reuse. And where you take GABA or glycine, break it down into glutamine to move it back through here. Or if it's just a pure glutamine neurotransmitter, they just grab the glutamine, stick it in here, and swoop it back up to get it reset. Right? So that's the reuptake process. You can sort of see there's very specific like neurotransmitters that have very specific enzymes that go with them that break it down and move it back in. So, how y'all doing? Feeling good? Did I go too fast or any of this? That makes sense. Okay, that's where we're stopping for today. Y'all can get on out of here. I'll see you back in on Tuesday. So have a good weekend. Let me know if you need anything, and take care.